Did you know some DNA survived for millions of years? Let's see if I can crack the code. Ancient DNA, or aDNA, is like a time capsule that allows us to peek into the lives of organisms that lived millions of years ago. By studying aDNA, scientists can learn about evolutionary history, how species adapted over time, and even the age of life on Earth. It's a pretty cool way to connect with our biological past. Preserving DNA over such long periods is incredibly challenging. Environmental factors like heat, light, and water can cause DNA to degrade quickly. Imagine DNA as a fragile piece of string that can easily unravel when exposed to harsh conditions. Even microorganisms like bacteria and fungi can feed on DNA, breaking it down into smaller pieces. Over time, these processes can reduce DNA into tiny fragments, making it difficult to recover. The rate of DNA degradation can be influenced by factors like temperature, pH levels, and the presence of oxygen. For example, DNA degrades faster in warmer and more humid environments. This is why DNA from ancient human remains is often found in colder climates, like in caves or permafrost. In these conditions, the DNA is more likely to survive for longer periods. DNA degradation is a complex process that involves the breakdown of its two strands. When DNA is exposed to water, the bonds between the nucleotides that make up the DNA strand can break, causing the strand to fragment. This process can be accelerated by the presence of enzymes that chop up DNA, like DNases. These enzymes are naturally present in many environments and can quickly degrade DNA. In addition to environmental factors, the way DNA is stored can also affect its preservation. For example, DNA stored in ice core samples or in dry conditions can last longer than DNA exposed to the elements. This is why scientists often look for DNA in places like permafrost, ice cores, or ancient sediments, where the conditions are more favorable for preservation. To extract and analyze ancient DNA, scientists use a variety of methods, including ice core samples, sediment analysis, and fossil remains. By studying these samples, researchers can identify the conditions that favor DNA preservation. For instance, DNA is more likely to survive in cold, dry environments with low levels of oxygen. In these conditions, the degradation process is slowed down, allowing DNA to last longer. Ice core samples are particularly useful for retrieving ancient DNA. These cores contain layers of ice that have accumulated over thousands of years, and they can provide a record of DNA from ancient organisms. By analyzing the DNA in these cores, scientists can learn about the history of life on Earth and how it has changed over time. Sediment analysis is another method used to extract ancient DNA. By studying sediment cores from lakes, oceans, and other bodies of water, researchers can identify the DNA of ancient organisms that lived in those environments. This can provide valuable insights into the evolution of species and how they adapted to their environments. The conditions that favor DNA preservation are often found in extreme environments, like permafrost or Antarctic ice sheets. In these environments, the cold temperatures and low levels of oxygen slow down the degradation process, allowing DNA to survive for longer periods. By studying these environments, scientists can gain a better understanding of how DNA can survive for millions of years. One of the most fascinating discoveries of ancient DNA is from the woolly mammoth. By analyzing DNA from frozen remains, scientists have been able to learn about the evolution and extinction of these iconic creatures. The DNA of mammoths has provided insights into their genetic makeup, their habitats, and even their interactions with early human populations. Ancient DNA from prehistoric plants has also provided valuable insights into the evolution of life on Earth. By studying DNA from fossilized plants, researchers can learn about the history of plant evolution, how they adapted to changing environments, and how they interacted with other organisms. The study of ancient DNA has significant implications for conservation. By learning about the genetic makeup of extinct species, scientists can gain a better understanding of how to protect endangered species today. This knowledge can be used to develop more effective conservation strategies and to preserve the genetic diversity of threatened populations. The study of ancient DNA continues to advance our understanding of evolutionary history and the age of life on Earth. With new discoveries and advances in technology, scientists are able to extract and analyze DNA from increasingly older and more degraded sources. This has opened up new avenues for research and has provided new insights into the history of life on Earth. To wrap things up, the survival of DNA over millions of years depends on a combination of factors, including environmental conditions, degradation processes, and the methods used to extract and analyze the DNA. By studying ancient DNA, scientists can gain a better understanding of evolutionary history in the age of life on Earth. If you're fascinated by ancient DNA and evolutionary biology, I'd love to hear your thoughts in the comments. What do you think is the most interesting discovery in the field of ancient DNA? Let me know. And if you're interested in learning more about evolutionary biology, be sure to check out my other video on the topic.